So we're going to begin with our first awardee, who is Susan Horwitz. She is Distinguished Professor, Rose C. Falkenstein Chair in Cancer Research, the Department of Molecular Pharmacology at Albert Einstein College of Medicine, New York. She received her award for defining novel mechanisms of action and resistance of drugs of natural product origin, most significantly Taxol, and promoting their use for treatment of cancer. And as has been said by many people, including people in this room, of course, Taxol has been a major, important drug in treating cancer, particularly breast cancer. But it was Susan's work in working out its mechanism of action that really gave the impetus to say this is doing something fundamental in cells, in inhibiting microtubules that could have importance for treating cancer. It's been a pleasure to meet and interact with Susan as we've traveled here, there, and everywhere, and it's wonderful to welcome her here to Toronto. Her lecture is called Nature as a Remarkable Chemist, the Discovery and Development of Taxol.
Good morning, everyone. If you cannot hear me, please raise your hand because you can't hear. Because it was a little difficult hearing Janet, and I want to be sure everyone can hear. It's a great honor to be here, to be a recipient of this year's Gairdner Foundation Award. And I'm delighted to tell you about the work that I've been doing that encouraged my acceptance of this award. So as you know, the title of my talk is Nature as a Remarkable Chemist, the Discovery and Development of Cancer. And this story goes way back to the 1960s when the Department of Agriculture and the National Cancer Institute got together and decided that they wanted to determine whether there were new natural products that could produce drugs that may be helpful in the treatment of cancer. And I have been interested in natural products and drugs for my entire scientific career, which is quite a few years. It turned out that the group of young students with a botanist by the name of Arthur Berkeley went out to the West Coast and started to look for just trees and twigs and leaves and brought these all back to the University of Wisconsin, where they would make extracts from this material and they would test to see if there was any effects on cells growing in tissue culture. And when they got a hit, they would then send that material to a medicinal chemist who was in contract with the NCI to try to isolate and purify the compound. And we are really very fortunate that this material went to two spectacular medicinal chemists by the name of the late Monroe Wall and Mansoor Gwani, who were able to isolate, and you must remember this is in the 1960s, this compound, and to determine its structure, which you will see is extremely complex. So this is a twig or a branch from Taxus brevifolia, the tree, and you can see here, this is called the Western yew, and this is from the bark of this tree that they were able to isolate Taxol, which I say is a very difficult task because the drug is present at only 0.0001% in the bark. But I want to tell you a little bit about the complexity of nature because it isn't just the bark from which the tree was isolated. In the bark grow endophytes, particularly fungi, and it's this relationship between the fungi and the bark which produce Taxol. And there is not a symbiosis, and the fungi does not cause disease in the tree, but together they are able to produce this drug. If you remove the fungi from the tree, it will not produce the drug. So what I want to emphasize is that we are all part of ecosystems which interact with bacteria, with fungi, with trees, and with sponges that grow in the ocean, all working together to produce molecules that really protect them. And you will see that most of these are trees, for an example, which are quite sessile, cannot run away from things which might attack them. So I want to remind you about the molecules that change the world, because most of us don't remember that when we go to the pharmacy and pick up our drugs, today in the United States and in Canada, about 50% of the prescriptions that we pick up are actually from natural products. And of course, aspirin being so well known, quinine, malaria, all of these, and you can really say that the pill has changed the world. Penicillin, which gave us each a 10-year increase in our lifespan. Many of us, I'm sure, are on lipid-lowering drugs. Vinblastine and vincristine were actually developed here in Canada at Western Ontario in the 1950s. And they were very important because it was thought that these drugs may actually decrease sugar in the blood and be useful for diabetes, but it turned out, of course, that these drugs are very useful in treating leukemias, Hodgkin's disease. And lastly, I have added Taxol. So how did I get involved with Taxol? Just by chance. I received a letter from the National Cancer Institute, you can see, in 1977, asking me if I would look at the mechanism of action of this drug. It had nothing to do with protein synthesis. And why did they write to me? I was an assistant professor. Well, I had already been working with a number of compounds, camptothecin, the etopicides, when I was a postdoctoral fellow, and then I had now been studying bleomycin extensively. They wrote me that letter. They didn't send me any money, but I liked the structure, and I'll show you that in a minute. So I had a new graduate student by the name of Peter Schiff. He was looking for a thesis project. I said, okay, we'll write to them and we'll ask them for 10 milligrams. And if at the end of the month it doesn't look like it's going to be a good thesis project, we'll have to look for something else. That month has never come. So I didn't know anything about the tree. I work in the Bronx at Albert Einstein, close to the wonderful Botanical Institute in New York, and I drove down there with Peter, and we said to them, do you know anything about this tree? And they said, yes, there's a great specimen at Greenwich, Connecticut at the church. So we drove to Greenwich. I didn't see any trees at the church. I went inside, and they said, oh, in the cemetery in the back. So I could give my whole lecture on the folklore of the yew tree, but I'm not going to do that. Just to tell you, go to the British Isles, you will see many yew trees in cemeteries. The yew tree is associated with death. And why? Because it's a very supple wood. And the bows and arrows that Robin Hood made were made from the yew tree. And they were so important in the English-French wars. So that the yew, if you read Shakespeare or T.S. Eliot, you see they talk about the yew. And even in modern literature, oh, I guess I omitted the next slide, they also talk about the yew tree. So what is Taxol and what does it look like? And it is a wonderful drug, a fabulous structure that nobody but a tree would make. Here you see this 6, 8, and fused 6-membered ring. This truly unusual 4-membered occitan ring. There are 2 side chains. The side chain at C13 is absolutely required. If you remove it, this is called bacatan, and it's totally inactive. So where does Taxol come today? Because, of course, Taxol is used in a large number of treatments for malignancies. When I started this project, there was exactly 1 paper in the literature, and that was the paper from Monroe Wall and Mansukhwani in JACS, the Journal of American Chemical Society, published in 1971. Today, there are 35,000 papers that have the word Taxol or its generic name, paclitaxel, in its title. And of course, the drug has now been given to millions of patients. So this is the drug that the tree makes. Abraxane is an important molecule. It's a nanoparticle that contains Taxol in it, the exact same drug, and it's preferred by many patients because it has less toxicity. Unfortunately, though, Medicare will not pay for that in the States, and the Canadian health system will not pay for that unless you have an allergy to Taxol. And these two molecules are derivatives, you can see here. This is a change here in this ring system, and this has been very useful in the treatment of breast cancer, and this is another derivative that's FDA approved, Taxol. So what Taxol does is it interacts with microtubules, which is part of the cytoskeleton, and a very important component of the microtubules that interact with hundreds of accessory proteins. And these are dynamic systems, which have to be dynamic to respond to changes in the cells. And you can see how many different things are required that Taxol can interact with and affect, because it inhibits the dynamic activity of the microtubules, such as mitosis, the ability of white blood cells to crawl, to change the shape of cells. Many activities need normal dynamic microtubules. So one of the early experiments we did was to just ask the question, does Taxol, which we could show in the laboratory was killing cells at 10 to the minus 9th molar, it's extremely active, we could show that all the cells, here you have a normal DNA, and DNA is replicated, but all of the cells here were blocked in mitosis. So to be honest, I thought we had maybe another vinca alkaloid. But I asked my student to look at the cells, always observe. We looked at these cells, and of course they were just filled with microtubules and bundles of microtubules. So it actually is an antimitotic agent, but I want to make the point that it does many things to cells, not only block them in mitosis, but has effects during the intercellular areas. Here's a normal cell, an interphase. You can see these beautiful red microtubules here. And after you treat this cell with Taxol, you can see that the microtubules exist in these distinct bundles. And this is very unusual, and this was fascinating to me. This was a new phenotype that we saw when we treated the cells with Taxol. If you have a patient that's on Taxol and you look at their white blood cells, you see these bundles because they are indicative of the activity of Taxol. So of course they have major effects during interphase cells, since microtubules, and you'll hear more about this, I know, are used for transport in cells, and for an example, for androgen receptors, for viruses, for kinesins. So this is a drug that has many different effects in cells. So one can isolate microtubules from calf brain we used to do. Now everybody just buys it, but we used to get the calf brains. The whole lab would work on isolating the microtubules. And you can see, you can actually watch the microtubules, the tubulin, the soluble tubulin, form a microtubule. And you have a lag period, you need GTP. And if you do this in the cold and then add cold or calcium, they completely depolymerize. Taxol is a remarkable drug. It wants to make polymers, and you can see here it has no lag period. This will happen in the cold without GTP. These are stable microtubules. If you add Taxol to a normal microtubule, it instantly stabilizes. So really, to me, the major question was, how is Taxol doing this? What is the binding site for Taxol, and how do we go about doing this? Not so simple, because Taxol doesn't make a covalent bond with the microtubule. So we decided that we would actually use photoaffinity labeling. I'll just show you how a microtubule is formed. You start with an alpha and beta dimer. They come together to make a protofilament, and this is the microtubule. It's a hollow tube. The betas come together, and the alphas come together, and this is a dynamic microtubule, which, because of dynamic instability, will actually depolymerize and then repolymerize. And what I want to point out one thing, that there are numerous isotypes of these alpha-beta tubulins, and that's something that I am extremely interested in studying, and I will tell you about this in a moment. So at this point, I was off to Hawaii to a Bleomycin meeting because I was studying Bleomycin. I met John Hartwell there, and John Duros, who were head of the National Cancer Institute, and I was excited. I was young. I said, this is a wonderful drug. And he said to me, well, when you get home, write me a letter. And I did. And I said, it's different from any other drug we've studied, and I plan to pursue it. And it would be really great if I could get a radio-labeled Taxol or some kind of label so that I could understand how it binds. I never got an answer, but that's okay. That meant I had to do it myself. And I had a young new graduate student by the name of Jerry Parnes, and he was able to put a radio-labeled tritium at the C7 position, which we knew from our work was for structure activity studies, is that you could add something to that system. And then we did an experiment, which is very weak, but showed us that all the label was here on the beta tubulin. So we were excited about that, but we realized if we were going to pursue this, we needed to get actually molecules that had radio-labeled and had photoaffinity analog so that we could study them. And this represents 10 years of work and 3 different molecules that we used. It wasn't easy to get organic chemists to synthesize these, particularly with tritium, because organic chemists don't like tritium. But I was very fortunate in enabling to get Professor Swindell, Kingston, and Ojima to make these molecules. These are the azeto compounds, which will activate in light, and this is the phenyl group here that will actually be activated, and then we have the tritium. We would interact these compounds with the molecules, with tubulin. We would shine the light. We would then do cyanogen bromide, pepsin, get the smallest possible piece with trypsin, and then do the old-fashioned N-terminal sequencing to find out where they were binding. And you can see here this bound to the beta 131, this to beta 217, 231, which is an important area. This is very rich in lysines, and we know this is an important area. And this actually, the beta Aj 282, which is in the M loop, which is a very important part when the beta tubulins come together. So I was working extensively on this, but what I didn't know that even Argolis was working in California in Ken Downing's lab doing something called electron crystallography. She was able to get a picture using tubulin and zinc sheets to actually get a fantastic picture of the alpha and beta tubulin and where Taxol binds. All of our photoaffinity work fitted beautifully to what she had shown. And it's interesting to note that this is a hydrophobic binding site here where Taxol enters. And in the alpha tubulin where it doesn't bind, there is an extra 8 amino acids. So there is no place in the alpha tubulin to get binding. So this is interesting because here you have the M loop, which the beta M loop interacts with the next beta with the H1S2 loop, and believe that the conformation changes in the presence of Taxol and that you get very strong interactions there, and that is what's stabilizing the microtubules. This is another way of looking at this. Here you have Taxol, but why I want to show this is the alpha and beta tubulin, and this is the H12 helix, and these interact with the many proteins that are in the cell, and we've been able to show that if you have Taxol here, these parts of the molecule will interact differently, sets for an example with something like MAP4. So Taxol has many effects in a cell, and remember that when you give a patient a drug, as Taxol is often given once a week, the concentration of drug is very different on day 1 than on day 6, so that the effects in the cell also can be very different. So what was going on at that time? Clinicians were totally uninterested in what I was doing, no interest whatsoever. As I said, I wrote to every drug company in the United States asking for help in labeling, didn't get any answers. But the molecular biologists were very interested because instantly they saw that they had a very good new tool to study the cytoskeleton. So the NCI saw a tremendous increase in requests for the drug. But we published our first paper in 1979. I always say when I'm talking to students, some of you weren't born. And this, I think, was very important in encouraging the NCI to move forward and to really push to try to get this drug out. So in preclinical toxicology, this Taxol is an incredibly hydrophobic agent, and actually the ability to get this into patients is very difficult. And so it was given in chemophore, which is actually a gamish, that's the best way to say it, of alcohol and castor oil, and that in itself has a problem. You give chemophore to a dog, and the dog totally loses its blood pressure. And in fact, we still don't know if some of the side effects are not due to the drug, but rather to the chemophore that's used. And also, the first patient that was given Taxol died of an anaphylactic shock. That would be enough to get rid of some drugs. But we got together, pharmacologists, oncologists, medicinal chemists, try to work this out. You can see there's a 5-year hiatus here. And during this time, it became clear that the best way to give Taxol was not as a bolus altogether as it had been given, but over 24 hours, and also to pre-treat the patients with steroids and antihistamines. And that's what made the difference. And then after that 5-year hiatus, when it went back into patients, it was clear that it had clinical activity. You can see in breast cancer, particularly in advanced drug-refractory ovarian carcinoma. And there was a big problem, which a lot of young people don't understand today. There was no patent. They had isolated the drug, Wani and Wall, in the 60s. We had done work, but nothing was patented. And there wasn't a drug company that wanted to pick it up. So the National Cancer Institute, which had done all the clinical trials, actually developed a crater, and it was put out, and Bristol-Myers Squibb picked it up, and they got 8 years of exclusivity for picking up the crater. To their credit, they got the drug on the market. They used the semi-synthetic method. Today, you isolate the major complex part of the molecule from trees in the Himalayas. Those needles will grow back, and then you put on semi-synthetically the side chain. So this continues with using Taxol in combinations. Even today, you're trying Taxol together with immunotherapeutics. So I assumed that Taxol was simply a drug that there would be many more coming along, which had the same mechanism of action. But we didn't see anything for 15 years. Taxol was a blockbuster drug. And when it became clear that there was money to be made, and there was, Bristol made $1.2 billion the first year it put it on the market. I didn't get a penny. People really started looking. So I want to show you a few of these because it's so interesting to me. These are different chemical structures, but they have the same mechanism of action. So the epophylones came isolated from a soil sample from South Africa that went to East Berlin, and they isolated the epophylones. You put a lactam on. You have ixabepilone, which is FDA approved for metastatic breast cancer. Discodermalide also is a stabilizing drug of microtubules, so I'm going to tell you a little bit more about this drug. And this comes from a sea sponge growing in the Bahamas. All three of these drugs bind at the same binding site in beta-tubulin, but not exactly in the same way. These two drugs, which have never had enough to test clinically, come from the sea in New Zealand and bind at another site, but in beta-tubulin. And we did a number of experiments in the lab, and we found that discodermalide could actually be quite active in cell lines that we had developed that were resistant to Taxol, and that intrigued us. And then we also saw that discodermalide and Taxol had synergistic effects in mice that had tumors. So that was unusual to us, and we actually wanted to study that, and I don't have time to tell you about it, but with that in mind, the discodermalide and the Taxol, I have been working extensively with Amos Smith, a professor at chemistry in Penn, and we are making many different analogs of discodermalide and hybrids with actually discodermalide and Taxol. So now I want to switch a little bit and tell you about the microtubule isotypes and why I'm so interested in them. We know that 50% of the women who get Taxol for ovarian cancer do not respond, and about 50% do. And we have absolutely no idea why this happens. So one thought is that perhaps a small part of this may be due to have something to do with the tubulin isotypes, and that maybe Taxol does not interact with all the isotypes equally, and the response to Taxol might have something to do with the isotypes that are in each tumor. And so that's the thinking behind my interest in the different microtubule isotypes. So there's a diversity of functions. There are 7, 8 actually now beta-tubulin isotypes. There are multiple genes I'll show you. The C-terminal peptide of all the isotypes are highly divergent, and that's how we name them, from the C-terminal peptides, and they are located on the surface of the microtubule, as I showed you. So this shows you the different beta isotypes, and it's this carboxyl end, negatively charged, that we call the isotype defining region. In addition, tubulin is highly post-translationally modified, and much of that is actually on these carboxyl ends. So here you can just see some of the polyglutamation. They're polyglycin elated. There's reversible tyrosination. There's acetylation, but this is at the alpha tubulin. But many of these are actually at that carboxyl end, that negatively charged carboxyl end, which define the isotypes. And that's of interest because those isotypes have quite a bit of information because the end is different, the post-translational modifications differ, and actually people talk about a tubulin code, which has a great deal of information on it. So this tells you something about the genes. I won't go through this, but to mention that they're all on different chromosomes. There's organ expression of these different isotypes, and there's also cellular expression. When we became interested in this, nothing was known about class 5, nothing about tissue specificity, and nothing about the cellular expression. So I had a new student in the lab, Susie Chow, and she decided to make a human antibody against beta-5. She made a superb antibody. In fact, it's too good because it doesn't interact with the mouse beta-5. But I'll just show you some of the things that she found when she looked at the distribution of beta-5 in normal tissue by immunohistochemistry. And the thing that really surprised us when we looked at this data was the fact that most of these were actually islets, ducts, sebaceous glands, things that are involved in secretion. And so the question became, is beta-5 specialized microtubule isotype looking at secretion? And that, of course, is interesting to know that perhaps the different isotypes have different activities in cells. In addition, when we actually looked at secretion in some tissues, you can see here that lights up the pancreatic duct, the bile duct, and very interestingly, normal breast duct. But when you have a lactating breast producing milk, you have a lot more of the beta-5 isotype. And interestingly also, it's very prevalent in certain tumors, and that the tumor can have different isotypes being present compared to the surrounding tissue. So we are interested, of course, in this isotype, and actually are looking now at fallopian tubes from humans, because if you know, fallopian tubes are thought to actually be involved in the development of ovarian cancer, and we see the beta-5 in the secretory cells of the fallopian tube. So lastly, beta-3 has been known for a long time, this is a paper from 2006, to affect the overall survival of tumors in advanced ovarian cancer, depending on a low or high versus beta-3 tubulin. So we were particularly interested in that, and we did some experiments. These are difficult experiments that Dr. Xiaoping Yang did in my laboratory. We developed an isoelectric focusing method that we can separate actually the different isotypes and different post-translational modifications. We can cut these out. We actually use mass spectrometry to determine exactly what they are. We're using a photoaffinity analog of Taxol. We have Tritium, and we can measure how much drug is associated with these different molecules. And I'll just show you here that beta-3 binds much less Taxol than does, say, beta-2. So we then looked very carefully at some of the sequences in the different isotypes. And we particularly looked at this region where I told you there's high leucines. It's called the area of, lysines. And there are 4 lysines here. But when we looked here at the sequencing, what you see here is that beta-3 has an alanine distinctly different from threonine. So with this information, I'm very fortunate to have wonderful colleagues at Albert Einstein, and I went to talk to Dr. Fisa, who does a lot of modeling and does also molecular dynamic solutions, molecular dynamic simulations, and he looked at both beta-1, the average distance between some of these various positions, and compared those to our variant at beta-3. And what you can see is that in many instances, such as in between positions 279 and 360, that the time that you have available for Taxol is much greater in the beta-1 than where we have the variant. Here again and here again. So our conclusion is that when you have that beta-3 variant with the change in the amino acid, that the binding site for Taxol is not as prevalent, it's not as available as much as it is with the other isotypes, which have a threonine there instead of the alanine, have the alanine. So again, this is a model. It may not be true, but it gives us ways to think about how Taxol interacts with different isotypes. And this, as I said, is an area that I'm particularly interested in now. So lastly, we are now trying to make human recombinant tubulins, which is not easy because you have to make the alpha and beta tubulin together. If you make just the beta tubulin, you do not get the right conformation for the Taxol. So we have to make these together, and we've been helped by Antonina Rolmakic, who's at the NCI, and you can see in the human recombinant beta-3 tubulin, we really get very little interaction with Taxol. And we're now working very hard to actually be able to have all of these human recombinant isotypes. So needless to say, this is the work of many, many different people. I would not be standing here today if it were not for the wonderful graduate students I've had, postdoctoral fellows, visiting faculty, and of course all my wonderful colleagues at Albert Einstein, whom I speak with, who have helped me over the years. And this award, which I'm so happy to receive, is really due to the work of many different people. Particularly, I want to mention Haley McDade, Cha Ping Yang, who have been with me for many years and who are my very best colleagues. Thank you very much.